Good morning, everyone, and thank you for this opportunity to present our work. I will be talking about how HSP70 in the dendritic cells modulates pancreatic cancer tumor microenvironment. I am Bhuvan Giri, and I am a general surgery resident at the University of Miami in Jackson Memorial Hospital program. So HSP70 is an inducible heat chaperone protein that has previously been shown to be associated with cancer growth, but that's almost exclusively in epithelial cancer cells. As you can see here, compared to normal ductal pancreatic cells, epithelial pancreatic cancer cells tend to overexpress HSP72. But our understanding of tumors as a combination of cancer cells, stromal cells, and a reactive immune infiltrate is constantly evolving. So we wanted to ask, what is the role of this protein in the surrounding tumor microenvironment? For this, we designed a simple experiment where we implanted pancreatic cancer cells into the wild-type mice or HSP70 knockout mice, thereby simulating a situation whereby cancer cells have HSP70, but only the tumor microenvironment does not have HSP70. When we looked at the tumors at endpoint, we saw that these tumors lacking HSP70, specifically in the tumor microenvironment, had barely any tumor growth. And this was true with not only pancreatic cancer models, but also in murine models of cancers like colon cancer, MC38, and the melanoma B16F10 model. Now, to begin somewhere, we can start by dividing the tumor microenvironment into stromal cells and the immune infiltrate. When we looked at the various stromal cell markers or specifically depleted HSP70 in the tumors, we did not see any difference in tumor growth. This data is not shown here for the interest of time, but when we looked at the tumor immune infiltrate, we saw that dendritic cells in HSP70 had an increased expression of anti-tumor molecules like TNF-alpha and MHC2. And this is true even in vitro system where there is an increased expression of MHC2 in HSP17 knockout dendritic cells. More interestingly though, injecting HSP70 knockout dendritic cells into wild-type tumor-bearing mice tended to decrease their tumor growth compared to only injecting wild-type dendritic cells. And although we were not able to reach statistical significance in this experiment, we show that even in this unclean system, HSP70 in DCs is able to modulate immune system response. Now, in conclusion, we show that there is a complex interplay of tumors and their immune microenvironment, that HSP70 in the dendritic cells play an important immunomodulatory role, and further research holds great promise in studying these in detail to design tumor vaccination strategies. Thank you for your time, and I would like to thank my mentors and my lab members for help with this project. Thank you.